Anatomy of flowering plants. You can very easily see the structural similarities and variations in the external morphology of the larger living organism, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. A study of internal structure of plants is called anatomy. Plants have cells at the basic unit. Cells are organized into tissues, and in turn, the tissues are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptations in diverse environments. 6.1 The tissues. A tissue is a group of cells having a common origin and usually performing a common function. A plant is made up of different kinds of tissues. Tissues are classified into two main groups, namely meristematic and permanent tissues, based on whether the cells being formed are capable of dividing or not. 6.1.1 Meristematic tissues. Growth in plants is largely restricted to specialized regions of active cell division called meristems. Plants have different kinds of meristems. The meristems which occur at the tips of roots and shoots and produce primary tissues are called apical meristems. Root apical meristem occupies the tip of a root while the shoot apical meristem occupies the distantmost region of the stem axis. During the formation of leaves and elongation of a stem, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in the axils of leaves and are capable of capable of forming a branch or a flower. The meristem which occurs between mature tissues is known as intercalary meristem. They occur in grasses and regenerate parts removed by the grazing herbivores. Both apical meristems and intercalary meristems are primary meristems because they appear early in life of a plant and contribute to the formation of the primary plant body. The meristem that occurs in the mature region of roots and shoots of many plants, particularly those that produce woody axils and appear later than primary meristem, is called the secondary or lateral meristem. They are cylindrical meristems. Fascicular vascular cambium, interfascicular cambium, and cork cambium are examples of lateral meristems. These are responsible for producing the secondary tissues. Following division of cells in both primary and as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or mature cells and constitute the permanent tissues. During the formation of the primary plant body, specific regions of the apical meristem produce dermal tissues, ground tissues, and vascular tissues. 6.1.2 Permanent tissues. The cells of the permanent tissues do not generally divide further. Permanent tissues having all cells similar in structure and function are called simple tissues. Permanent tissues having many different types of cells are called complex tissues. 6.1.2.1 Simple tissues. A simple tissue is made up of only one type of cells. The various simple tissues in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma forms the major component within organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal, or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have small intercellular spaces. The parenchyma performs various functions like photosynthesis, storage, and secretion. The collenchyma occurs in layer below the epidermis in most of the dicotyledonous plants. It is found either as a homogeneous layer or in patches. It consists of cells which are much thickened at the corners due to a deposition of cellulose, hemicellulose, and pectin. Collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplasts. These cells assimilate food when they contain chloroplasts. Intercellular spaces are absent. They provide mechanical support to the growing parts of the plant, such as young stem and petiole of a leaf. Sclerenchyma consists of long, narrow cells with thick and lignified cell walls having a few or numerous pits. They are usually dead and without protoplasts. On the basis of variation in form, structure, origin, and development, sclerenchyma may be either fibers or sclerates. The fibers are thick walled, elongated, and pointed cells, generally occurring in groups in various parts of the plant. The sclerate are spherical, oval, or cylindrical, highly thickened dead cells with very narrow cavities, lumen. They are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear and sapota, seed coats of legumes, and leaves of tea. Sclerenchyma provides mechanical support to organs. 6.1.2.2 Complex tissues. And the complex tissues are made of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissues in plants. Xylem functions as a conducting tissue for water and minerals from roots to the stem and leaves. It also provides mechanical strength to the plant parts. It is composed of four different kinds of elements, namely tracheids, vessels, xylem fibers, and xylem parenchyma. Gymnosperms lack vessels in their xylem. Tracheids are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and are without protoplasm. The inner layers of the cell walls have thicknings which vary in form. In flowering plants, tracheids and vessels are the main water transporting elements. Vessel is a long cylindrical tube-like structure made up of many cells called vessel members, each with lignified walls and a large central cavity. The vessel cells are also devoid of protoplasm. Vessel members are interconnected through perforation in their common walls. The presence of vessels is a characteristic feature of angiosperms. Xylem fibers have highly thickened walls and obliterated central lumens. These may either be septate or aseptate. Xylem parenchyma cells are living and thin-walled and their cell walls are made up of cellulose. They store food materials in the form of starch or fat and other substances like tannins. The radial conduction of water takes place by the ray parenchymatous cells. Primary xylem is of two types, protoxylem and metaxylem. The first formed primary xylem elements are called protoxylem and the later formed primary xylem is called metaxylem. In stems, the protoxylem lies towards the center, pith, and the metaxylem lies towards the periphery of the organ. This type of primary xylem is called endarch. In roots, the protoxylem lies towards periphery and metaxylem lies towards the center. Such arrangement of primary xylem is called exarch. Phloem transport food materials, usually from leaves to other parts of the plant. Phloem in angiosperms is composed of sieve tube elements, companion cells, phloem parenchyma, and phloem fibers. Gymnosperms have albuminous cells and sieve cells. They lack sieve tubes and companion cells. Sieve tube elements are also long, tube-like structures arranged longitudinally and are associated with the companion cells. Their end walls are perforated in a sieve-like manner to form the sieve plates. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks a nucleus. The function of sieve tubes are controlled by the nucleus of companion cells. The companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. The sieve tube elements are and companion cells are connected by pit fields present between their common longitudinal walls. The companion cells help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated, tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connections exist between the cells. The phloem parenchyma stores food material and other substances like resins, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fibers or bast fibers are made up of sclerenchymatous cells. These are generally absent in the primary phloem but are found in the secondary phloem. These are much elongated, unbranched, and have pointed needle-like apices. The cell wall of phloem fibers is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute, flax, and hemp are usually used commercially. The first formed primary phloem consists of narrow sieve tubes and is referred to as protophlloem, and the later formed phloem has bigger sieve tubes and is referred to as metaphloem. 2. The tissue system. We were discussing types of tissues based on the types of cells present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would also be dependent on location. On the basis of their structure and location, there are three types of tissue system. These are the epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. 6.2.1 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cells, the stomata, and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which forms a continuous layer. Epidermis is, Epidermis is usually single-layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cells, which enclose a stomatal pore. In grasses, the guard cells are dumbbell shaped. The outer walls of guard cells away from the stomatal pore are thin, and the inner walls toward the stomatal pore are highly thickened. The guard cells possess chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells are together called stomatal apparatus. The cells of epidermis bear a number of hairs. The root hairs are unicellular elongation of the epidermal cells and help absorb water and mineral from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. 6.2.2 The ground tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary range, in the primary stems and roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called mesophyll. 6.2.3 The vascular tissue system. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. In dicotyledonous stem, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues, and hence are called open vascular bundles. In the monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissues, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoined type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoined vascular bundles usually have the phloem located only on the outer side of xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization of roots, stem, and leaves, it is convenient to study the transverse sections of the mature zones of these organs. 6.3.1 Dicotyledonous root. Look at figure 6.6a. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cells of epiblema protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water-impermeable waxy material, suberin, in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis, such as pericycle, vascular bundles, and pith, constitute the stele. 6.3.2 Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. As compared to the dicot root, which have fewer xylem bundles, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. 3.3 Dicotinal System. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem. Covered with a thin layer of cuticle, it may bear trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of chlorine cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains, and the layer is also referred to as starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semi-lunar patches of Sclerenchyma. In between the vascular bundles, there are a few layers of radially placed parenchymatous cells which constitute medullary ridge. A large number of vascular bundles are arranged in ring. The ring arrangement of vascular bundles is a characteristic of Dicot stem. Each vascular bundle is conjoined, open, and with enlarged protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. 6.3.4 Monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath and a large, conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent, and water-containing cavities are present within the vascular bundles. 6.3.5 Dorsiventral dicotyledonous leaf. The vertical section of a dorsiventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface, adaxial epidermis, and lower surface, abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possesses chloroplasts and carry out photosynthesis, is made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at figure 6.8a and find the position of xylem in the vascular bundle. 6.3.6 Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermal cells along the veins modify themselves into large, empty, colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curl inwards to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles, except in main veins as seen in vertical section of the leaves. 6.4 Secondary growth. The growth of the roots and stems in length with the help of apical meristem is called the primary growth. Apart from primary growth, most dicotyledonous plants exhibit an increase in girth. This increase is called the secondary growth. The tissue involved in secondary growth are the two lateral meristems, Vascular Cambium and Cork Cambium. 6.4.1 Vascular Cambium. The meristematic layer that is responsible for cutting off vascular tissues, xylem and phloem, is called vascular cambium. In the young stem, it is present in patches as a single layer between the xylem and phloem. Later, it forms a complete ring. 6.4.1.1 Formation of Cambial ring. In Dicot stems, the cells of cambium present between primary xylem and primary phloem is the intravascular cambium. The cells of medullary ridge adjoining these intravascular cambium become meristematic and form the intervascular cambium. Thus, a continuous ring of cambium is formed. 6.4.1.2 Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cells both towards the inner and the outer sides. The cells cut off towards pith mature into secondary xylem, and the cells cut off towards periphery mature into secondary phloem. The cambium is generally more active on the inner side than on the outer. As a result, the amount of secondary xylem produced is more than secondary phloem and soon forms a compact mass. The primary and secondary phloem get gradually crushed due to the continued formation and accumulation of secondary xylem. The primary xylem, however, remains more or less intact in or around the center. At some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial directions. These are the secondary medullary ridge. 6.4.1.3 Springwood and autumnwood. The activity of cambium is under the control of many physiological and environmental factors. In temperate regions, the climatic conditions are not uniform throughout the year. In the spring season, cambium is very active and produces a large number of xylem elements having vessels with wider cavities. The wood formed during this season is called springwood or early wood. In winter, the cambium is less active and forms fewer xylem elements that have narrow vessels, and this wood is called autumn wood or late wood. The spring wood is lighter in color and has a lower density, whereas the autumn wood is darker and has a higher density. The two kinds of woods that appear as alternate concentric rings constitute an annual ring. Annual rings seen in a cut stem give an estimate of the age of the tree. 6.4.1.4 Heartwood and sapwood. In old trees, the greater part of secondary xylem is dark brown due to deposition of organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the central or innermost layers of the stem. These substances make it hard, durable, and resistant to the attacks of microorganisms and insects. This region comprises dead elements with highly lignified walls and is called heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and is known as sapwood. It is involved in the conduction of water and minerals from root to leaf. 6.4.2 Cork cambium. As the stem continues to increase in girth due to the activity of vascular cambium, the outer cortical and epidermis layer get broken and needs to be replaced to provide new protective cell layers. Hence, sooner or later, another meristematic tissue called cork cambium or phloem develops, usually in the cortex region. Phloem is a couple of layers thick. It is made up of narrow, thin-walled and nearly rectangular cells. Phloem cuts off cells on both sides. The outer cells differentiate into cork or phloem, while the inner cells differentiate into secondary cortex or phloderm. The cork is impervious to water due to suberant deposition in the cell wall. The cells of secondary cortex are parenchymatous. Phloem, phloem, and phloderm are collectively known as periderm. Due to activity of the cork cambium, pressure builds up on the remaining layers peripheral to phloem, and ultimately these layers die and slough off. Bark is a non-technical term that refers to all tissues exterior to the vascular cambium, therefore including secondary phloem. Bark refers to a number of tissue types which periderm and secondary phloem. Bark that is formed early in the season is called early or soft bark. Towards the end of the season, late or hard bark is formed. At certain regions, the phylogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cells. These parenchymatous cells soon rupture the epidermis, forming a lens-shaped openings called lenticels. Lenticels permit the exchange of gases between the outer atmosphere and the internal tissue of the stem. These occur in most woody trees. 6.4.3 Secondary growth in roots. In the dicot root, the vascular cambium is completely secondary in origin. It originates from the tissue located just below the phloem bundles, a portion of pericycle tissue, above the protoxylem, forming a complete and continuous wavy ring, which later becomes circular. Further events are similar to those already described above for a dicotyledonous stem. Secondary growth also occurs in stems and roots of gymnosperms. However, secondary growth does not occur in monocotyledons. Viewers, I think this audiobook would have helped you in revising the NCERT. If it has helped you, then please give your precious 10 seconds to subscribe my channel. Thank you very much.